Okay, I wanted to start out today with just a short presentation, just that is very similar to some things we've done before, but it has to do with the issue of electron microscopy. The reason this is so relevant and important is, as everybody I think knows by now, the only way that something the size and morphology and complexity of a virus has ever been seen is through an electron microscope. Anybody who says they've seen a virus under a light microscope is just flat out incorrect, because viruses are way too small to see under the magnification of a light microscope. And I've talked about the problems with electron microscopy images. And this is something that Hillman talked a lot about. And he actually came to the conclusion that every single thing you see under an electron microscope is an artifact. And that's because the steps that you needed to take in order to get this image includes, we're talking about biological samples or living specimens. First of all, you obviously have to kill it, and then you have to dehydrate it, and then you have to macerate it, and then you either have to freeze it like modern electron microscopy, or dye it, or there's all kind of processes, and I've been over those, and you can see all the steps. His point was that in order to say that the structure, and that's what we're talking about, the physical structure that you see in an electron microscope is identical or even reflective of something that's seen in real life. In other words, while the organism was living, would require a lot of control experiments. And some of them, frankly, as he pointed out, can't be done. So you can't control for whether killing the organism affects the appearance of the final structures you see under an electron microscope, because there's no other way to do it. So that's something you can't control for. You can control for some of the processes, but not all of them. You can't control for shooting an electron beam at a specimen, which immediately changes the nature of it. It dehydrates it, it changes the energetics. And obviously you can't control for that because there's no other way to see that image but besides in that technique. But some of the things he was able to control for, like certain reagents and certain stains, and he found that if you saw something that was the same, or depends on the situation, different than what you got with that stain, that means the stain irrevocably, irreparably changed the image, and you can't rely on the final image to be reflective of what you would see in nature. Just a simple example. Also, the other issue has to do with the location of what you're seeing. And again, if you realize that you took this tissue and you macerated it, which is similar to putting it in a blender, that would be similar to you take your old car to the dump or whatever, the car graveyard, and the first thing they do is drop that big concrete ball on it and smash it to smithereens. Now, if you had never seen or were never able to see an actual car, and that's the first car you saw, and you go to the car graveyard and you see a car with a steering wheel in the trunk, you might end up with a theory about how you can drive a car with a steering wheel in the trunk. And that would be based on your observations, but it would be an observation of a obviously extremely contrived situation, because the reality is steering wheels, while the car is functional, don't have their steering wheel in the trunk, at least not any car that I've ever seen. So that's just an example of how you can be fooled by this. And the reason this comes up with virology is because it's, if not one of the proofs, it's one of the main proofs that all virologists use to prove the existence of such and such a virus like SARS-CoV-2. So I thought I would take the opportunity to actually look at some electron microscope images and see what we can see. Now, these images are going to be taken from one of two places. Either they're all from peer-reviewed scientific journals. Some of them, and I'm not going to tell you how many, are from journals that are saying that these are electron microscopy images of SARS-CoV-2. Other images are taken from other journals that show you similar, you'll see, images, but most of these, I think all of them, were taken before the year 2000 in people who couldn't possibly have COVID or SARS-CoV-2 virus, and they were taken from either kidney biopsies or from lung biopsies or lung bronchial alveolar lavage fluid. In other words, some of them, there's no possible way they had SARS-CoV-2, and some of them, they're claiming that these are images of SARS-CoV-2. So if what the virologists are claiming is correct, that they can look at an image and say, yes, this is SARS-CoV-2, let's see how you people who are not virologists, let's see how you do with this. So I'm going to screen share. And here we go. Whoops. Okay. So again, this image that I'm going to show you is either SARS-CoV-2, the virus, or it's from a kidney biopsy or bronchial alveolar lavage fluid from before 2000. So nobody had COVID. Nobody says these are SARS-CoV-2. They're clearly not. They're just stuff in either the kidney biopsy or the bronchial fluid. Now, the first one I'm going to show you is, I'm going to give you the answer to this one, is SARS-CoV-2. And there you see the picture of it. You see the dots, which are meant to represent the spike protein. You see an approximate measurement of it, 100 nanometers. And there you go. So that's the reference. Okay, these ones I am not going to tell you because I'm hoping to show this to an expert virologist or electron microscopist someday and ask them to tell us, since this is one of the evidence that this thing exists, which ones of these are SARS-CoV-2 and which ones are just cellular debris. So what about this one? Take a look. You might want to vote as we go along. And then the next one is this one. You can see the arrow in the lower left-hand corner. And there's some other things that are similar in other places. And don't cheat. Don't look at the tissue. That's not what we're looking at. We're looking at the structure, which we're told represents the virus. Here's a bunch. They don't have arrows, but you can see 1, 2, 3, 4, 5, 6 or so different structures with the black dots, circles, all the rest of it. Here's another one, more of a close-up. This doesn't have a measurement, but they're all around the same size. Here we have a few more. And then there's some more. We have some arrows here, so you can see which ones they're referring to. It doesn't have a size measurement. And then I put 4, I just put them in the same slide just to make it a little easier. You can see the arrows pointing to the different things they're pointing out as either SARS-CoV-2 or debris from the kidneys or lungs. And again, you can see the measurements and the shapes, etc. So, and there you see in the lower left-hand corner, you see a close-up of one of them. So the question is, how many did you get right? Now, I'm going to be mean and not tell you the answer here because I'm hoping to show this to, as I said, some virologists or electron microscopists and see how many they get right. And I think that would be very interesting. Now, this isn't the only area where electron microscopy is problematic. And again, we're talking about finding structures which are real and not artifacts. Artifacts meaning they come as a result of the process of making the slide, right? We're not talking about whether, so here I'm going to focus on the sodium-potassium pump. And let's be clear, I'm not talking about whether sodium and potassium are found in the distribution in and out of the cell that we're told. That I don't dispute because it's measured. Nor do I dispute the critical importance of the fact that cells, mammalian cells, human cells, concentrate potassium inside the cell and exclude sodium. That's how they develop a charge across the membrane, which allows the cell to be alive and, quote, do work. So nobody is disputing that there is a unequal distribution, i.e. a concentration of potassium inside and sodium outside. And I would say that nobody even disputes that there may be a protein involved in this process. And I would even say that I wouldn't even necessarily dispute that ATP, which is erroneously referred to as the energy molecule, has something to do with this process. The question we're asking is there a sodium-potassium pump in the membrane? Basically a lipid protein structure, an actual structure like a virus or some other structure that is responsible for this. Now, this is one of the crowning achievements in modern biology and in medicine. There's actually a lot of drugs like digitalis that are based on affecting this sodium-potassium pump. The proteins have been supposedly elucidated and characterized. And again, nobody is disputing that there may not be a protein involved here. The question is it embedded in the pump? And the way you learn this is it's some sort of like a whirligig that binds to the potassium on the outside and then flips around and deposits the potassium on the inside of the cell, at the same time picking up the sodium on the inside, that's a different receptor, and then swinging around again like a whirligig sort of thing, and then depositing the sodium on the outside. And this, of course, takes energy, and the energy is supplied by ATP. If you want an image of this, I remember when I was a child going to New York City with my parents, and we went to a luncheon, and there was a counter with a bunch of sandwiches, looked like they'd been in there for a few weeks, that went around on a conveyor belt, and you put a quarter in at that time, probably $10 now, and then you could stick your hand in, open up the thing, and get the sandwich out. So that's sort of what we're talking about. So we're looking for that structure embedded in the cell membrane, and thank God, here it is. The problem, of course, is this is a drawing, and there is no such thing that looks like this, and that is a schematic drawing of a lipid bilayer cell membrane, which probably also doesn't exist. And there's the sodium potassium pump embedded in it, and you can see all that it does. So I said, I got to find a better image of this. So I found different kinds of receptors. This receptors in membranes, whether it's calcium gated channels or acetylcholine receptors or ion channels or G protein coupled receptors. That's one of the biggest issues in modern medicine and biology. That is how we say things get into the cell. So here's some pictures of them. The one on the left looks like a 4. The one on the right, the middle looks like a Y. The one on the right looks like a little y. And you can see the whirligig thing, and it turns around. But obviously, this is still a picture, a drawing, or computer-generated image, not a real receptor. So I continued in my search on the internet and looking through PubMed and papers. I wanted to see a real image. Well, the next thing I came up with was an X-ray crystallography image. So this is an X-ray crystallography picture of the sodium potassium pump. And I didn't realize this, but in order to do an X-ray picture, you have to totally dry the specimen. It has to be desiccated, no water. And obviously, if it's a protein, which are always embedded and shaped by the water, this cannot be the real image for that structure. But anyways, this was the X-ray crystallography picture of the sodium potassium pump embedded in the membrane, and you see the blue part in the membrane and the little tails on the inside and the outside, which pick up the sodium and potassium. But again, obviously, this is not a real image. This is a drawing or a computer-generated image. So I kept my search going literally for hours. I got to see a real image. And finally, I did. Here's an electron microscopy image of the sodium potassium pump. You can see the arrows. So I don't know about you, but actually interesting, looks like there's a SARS-CoV-2 at the top, right between the P's. I don't think this person had COVID. This was from years ago. But there's the image, a little black dot. Now, what's so interesting is how you go from a little black dot, which is the actual structure you see, which is, of course, a dot of stain, either osmium, I think, or some metallic stain that is dotting this, whatever kind of tissue this is. Again, how you go from that to this or this, frankly, is beyond me. Let me show you another one that has to do with the problem with electron microscopy. Again, I've shown these before. So we've all heard of another sacred cow, neuroanatomy, neurology 101, is that nerves have synapses in them, which are like moats. So if you took a nerve, it has an axon, sorry, it has a nerve body, the cell body, and that's where the impulse starts, and then it goes down a long tube called an axon, and then it ends at a synapse, and here you see the synapse, and the synapse has little vesicles which store the neurotransmitter like acetylcholine or serotonin, and when the nerve reaches the synapse, it stimulates the synapse to release its contents, which are these neurotransmitter chemicals, and then they swim across the moat and attach to receptors on the other side and start the next nerve going. And I've written and talked a lot about why I thought that process is way too slow to be realistic. But here I'm just looking for, my quest that morning was to find actual pictures. Does this structure, this anatomical structure called a synapse, actually exist? Again, the problem is it's too small to see under any light microscope that we know of. So you can't use light microscopy to find it. You have to do electron microscopy. But anyways, this is the first image I found, and there you see the two, the moat and the synapse, and the little the flashes of neurotransmitter crossing the junction. And again, obviously, this is a computer-generated image or a drawing, and it's not real. So I kept looking. Now, here's a better picture. Again, you see the moat. That's the sort of middle part. And then you see it looks red on one side and purple on the other. And it looks like the purple side is the presynapse that's swimming. The chemicals are swimming to the receptors on the other side. The problem still, this is again not a real image. It's a computer generated or drawing or something. So, and you may think I'm gaming the system, but I tried to find, okay, let's find an electron microscope picture that convinces me that there is such an anatomical structure. And there it is. I don't know if you can see right in the middle. I don't know. Yeah, my cursor works. There it is. SY. I don't have the code, the key here, but there is the synapse. I don't know about you, but that doesn't really convince me that that is anything like that or that. That looks like something that was caused by a dehydration of the tissue, and therefore it pulls apart, because you can never actually see a synapse in a live dissection of a nerve. But if you dehydrate it in a way to prepare the electron microscope pictures, then you see things pull apart, and then you see gaps, and you see the staining on either side of the gap. That's just normal how things stain. And then you end up with this picture, which supposedly represents a synapse, which I would contend doesn't actually exist.